So I'm going to start by talking about what microgels are and the big picture of my project. So microgels are just colloidally stable spherical hydrogels, and hydrogels are defined as water-containing polymer networks. These particles are anywhere from 100 nanometers to 1 micrometer in diameter, and they're a really unique class of particles because they have both stability due to their cross-linking in their chains, which you can see in this illustration at the top, but they also have fluid-like properties due to their really high water content. So in this picture, you can see these long lines represent the polymer chains and the dots represent the cross-linkers joining them together in both the hydrogel and microgel illustration. So commonly, we use poly-N isopropyl acrylamide, also called NiPAM, which is this molecule here, as our monomer, and this is a thermoresponsive molecule. And then we also use N,N-methyl bis acrylamide, or BIS, as a cross-linking agent. So these are a really unique class of particles because they possess the shape of a sphere, but their softness allows them to be super deformable. So they can do interesting tasks like fitting through pores smaller than their own size, or they can spread to have a diameter larger than their size. And the reason for their ability to change shape like this is, like I said, they're mostly made up of water. So the particle that I'm making is designed to specifically find and bind to blood clots in the body and then release tissue plasminogen activator, or TPA, and that breaks down the blood clot in the body, which you can see in this figure here. This is designed for a medical application specifically for patients who experience too much blood clotting. So this can also be used in something like what we see with a lot of COVID patients who have multiple organ failure due to clotting in their capillaries. This would be for an application kind of like that. So now that we have a big picture idea of what microgels are, let's look at some ways they're used and how we can implement those functions into the particle that we are making here. Okay, so the main theme of this research group is to use microgels to treat issues in thrombosis or blood clotting. And one example of this is using ultra-low cross-linked particles. So these are also called ULCs, and they can act as artificial platelets. And these are microgels that don't use an external cross-linker. So the only cross-linking we see is what's referred to as self-cross-linking between the polymer chains, which makes them like super, super soft and deformable. These ULCs have fibrin-specific antibodies bound to them so that they will bind to fibrin that the body releases upon trauma. So in this figure, you can see that the platelet-like particles are inactive until they bind to fibrin, which activates them. From here, they're able to deform and spread out and bind to more fibrin, and then eventually collapse to make a clot, which is what you see happening here. So although these particles are used to break down or to make clots and our particles are used to break up clots, we can use this previous work that's shown that ULCs interact better with clots than cross-linked particles, and we can implement that into the design of our own particle because we want something that will specifically bind to fibrin and that will interact well with clots in the body. Okay, so another study that's been done previously is this one, which shows drug release using microgels. So in this one specifically, core shell microgels were studied to have like a densely cross-linked core and a looser cross-linked outer shell surrounding it. And then with this design, the drug diffuses through the porous outer shell into the bloodstream. And in this specific study, it was TPA, which is what we're using. And the release rate of the TPA from these microgels depends on the level of cross-linking because the level of cross-linking dictates the pore size. So dextran dye was used to determine optimal pore size in these particles. And you can see in the figure that the 3 kilodalton dye was found mainly in the dense core, and the 70 kilodalton dye was found in the looser outer shell. So once they found that the 2% cross-linked outer shell was best for releasing dye, they then went on and did studies with the release rate of TPA using confocal microscopy to figure out the rate of diffusion of the TPA out of this particle. So from this, we're using the information that the 2% bis cross-linked shell is the best for the release rate of TPA, which is what we're going to be releasing as well. Okay, so using all this, we can then look at the particle that we're making. So as you can see from the figure, a densely cross-linked core is first synthesized using 10% bis as the cross-linker, and it also contains an azide on the cores, which is used for binding a dye to the particle for imaging, which I'll talk about a little more later. And then the first shell is synthesized to have a 2% bis cross-linking shell that encapsulates the TPA and releases it. And then finally, the second and outermost shell is the ULC with the fibrin-specific antibody bound to it to interact and specifically bind to the clots in the body. So this is the idea behind the design of the particle, and now we can talk about how I actually made it and imaged it. Okay, so here we have the synthesis broken down into its 3 different parts, and it's important to know that changing the mechanics and the functions of a microgel always happens at the synthesis step. So optimizing this is really important in getting the particle that we want. Each step is synthesized by a simple 1-pot free radical precipitation polymerization using NiPAM, our monomer. And this reaction is initiated by a persulfate initiator that decomposes into free radicals at high temperatures. And this free radical then reacts with monomers in solution to form polymer. So for the core here, we have the NiPAM monomer and the bis crosslinker initiated by ammonium persulfate to make a cross-linked core. We can then add GMA, which can react with sodium azide here to open up this ring and give us an azide attached to the core. We then image this core with atomic force microscopy, which you can see in this top left image. And this topographic image of the cores gives us information about the size of the particles. So what we see here is that the particles are all around the same size and shape, which indicates successful polymerization of the cores. And then from here, the next step is to take these cores and react them again to make core shell particles. So we're doing this again with NiPAM and bis and acrylic acid as a co-monomer. So we can see in the AFM images that the core shell particles are slightly bigger than the core particles, and we see again that size uniformity that we really like. And so then after characterizing these, we can finally react them one more time with the NiPAM monomer and acrylic acid and no external crosslinker. Because they are ULCs, like I said. And then the bottom AFM image is of the core shell shells, which appear larger in size than the core shells as we expect. And what's cool about this image is when you look closely, you can actually see that faint outer ULC shell on this image. And again, we see the size uniformity and shape that we like, which is really important. I want to highlight this in synthesis because when you're designing something that's going to go in your body, you want to make sure that you're going to get the product that you want and that there's consistency and predictability in the product. Okay, so this slide, I just wanted to talk about some of the challenges in synthesis. So this is an image I took when I first tried to synthesize my core shells, and it didn't exactly go as planned. But that's okay because we get some interesting information about why optimizing synthesis is so important from stuff like this. So here in the image, you can see that these larger particles are the core shells successfully synthesized, but then you also get all this heteronucleation that is shell particle that reacted with itself. So we need to get the correct ratio of core to shell so that the shell particles are statistically more likely to bind to the core than to themselves. So we can avoid this heteronucleation we see here. So once you do optimize that and do get the product like we did, then we can add dye to our clickable cores, which was the next step that we did. Adding a fluorophore to the core of the particle allows it to be tracked in the body in order to see the way in which particles flow through the bloodstream. And this is super important if you're releasing a drug from these particles by diffusion because you want to know how fast these particles are moving through the bloodstream and how that compares to the rate of diffusion of the drug out of the particle. So this click reaction you see here is actually super interesting because initially these types of reactions with an azide and an alkyne, which is the triple bond between 2 carbons, were only done with terminal alkynes, which is like at the end of a molecule. And they were done with a copper catalyst, but copper is super toxic to the body, so we can't use that for our application. It's not safe. So instead, this strained alkyne you see here pushes the molecular orbitals towards the transition state, which allows this reaction to be carried out without a catalyst. And this type of clickable core is used for tracking, and it's also really useful because it's a chemo-orthogonal functional group, meaning that it's not going to react with any of the proteins in the blood. And this allows us to attach fluorophores, which fluoresce near IR and creates good contrast imaging even in the body because tissues in the body do not fluoresce at this frequency. So here to the left is the confocal image of the cores the clickable cores once we added it to our final product. And this just confirms that we were able to make that core, click the dye to it, and that was successfully synthesized, and we can use that in future work. So to conclude everything, we made core shell particles. We imaged them using AFM to confirm that they look how we want them to look. And moving forward, the next steps would be, like I talked about in the beginning, doing loading and release studies of TPA, and you can also use dextran dye for this, like the other experiment showed. And then we would also go ahead and track these particles in the body, see how they move through the bloodstream and how they interact with clots in the blood. Okay, so I'd like to thank Dr. Lyon, Dr. Islam, and Sonika Pandit for helping me with this project, and funding by the National Institutes of Health. And that's it. So I'm going to open it up to questions now from everybody.
Thank you so much. All right. Thank you for the applause. I will ask for applause after the questions as well, but no problem with double the applause. So let me see. Please put your questions in the chat box, or feel free to unmute now as well. Let me pause in case anybody wants to unmute with a question. All right. Dela, I'm not sure if you're seeing one in the chat from Jordan. What was your motivation for joining Dr. Lyon on this project?
Well, I've been in this research group since sophomore year, actually. So I was interested in the lab at first because I wanted to use chemistry to approach like medicine in a way, and that was a way to do that. For this specific project, we were looking at, like, when COVID first started, we were kind of looking at how TPA could be used to break down clots, because like I said, that was an issue with a lot of patients. So that was interesting to me, and that just opens it up to a whole broader application of like taking what we already know about microgels and applying it to something different. So that's kind of what was the motivation behind this.
Great. I have a question as well. Your dye molecule, sorry about my dog. Your dye molecule is really large and interesting. I like the cyclooctane part. I'm curious, how did you choose that particular dye molecule? Like, what was it? Is there a particular color you're looking for it to have that's good for the body? How do you choose that?
This is what we've used previously. There's been other clickable core studies, so this has always been like our dye choice. And like I said, it's good because it won't react with anything else in the blood, and you don't have to co-synthesize with a dye this way with a clickable core. So there are like other dyes you can use out there, but this one just worked really well because, like I said, the strained alkyne makes it react really well, and we can do it after synthesizing. It doesn't mess up our synthesis, and it'll fluoresce really well in the body. So that was kind of the motivation.
Nice. Perfect. Any other questions for Dela?
Yeah, I had a question. I thought the research was really interesting, and I liked the part about microgels being able to fit in incredibly small pores, like you say, because they're very flexible, being mostly composed of water. I was just curious, I know you said you had some struggle cross-linking. What would you take from this synthesis into other synthesis if you were ever to go into research in the future? Like, what did you learn from working in the lab?
So you're probably talking about this. What I learned is that, like, although these syntheses are relatively simple to come up with theoretically, carrying them out is not always the same. So it's like, I think maybe it was you, Dr. Lieberman, who was saying, like, sometimes research is a little glorified to be this great, amazing thing, and you find these cool things, which is true, but there's a lot of struggle with it. So from this specifically, I just noticed how, like, my core to shell ratio was not where it needed to be. So that's where you kind of, and it can be difficult because you synthesize a core and you don't exactly know how concentrated it is. because they're pretty highly concentrated, but you don't know, like how many core particles you actually have. So it's a little bit of like a trial and error sort of thing. But once you optimize that, it's pretty easy to make it repeatable and reliable. It's just getting there. That's the hard part.
Yeah, that was really good. I was really impressed.
Thank you.
All right, any time for one last question if anybody has one? I don't see any more in the chat. If not, please join me in congratulating Dela on an excellent talk. Feel free to come off mute and make some noise with me. Woo!
Go, Dela!
Great job, Dela.
And thank you so much.
Go, Dela!
Thank you.
All right, let's see. Jordan, can you get your screen share started?
Yes, let me see if I can get it to come up. Can you see that?
Yes, and
right.
can you see us as well as you're presenting for me to use an emoji at the 10-minute marker?
Yes, I sure can.
Okay, fantastic.
For me, actually, I was going to put my virtual background on, but whatever, just ignore. Focus on me. Forgot to put that on.
We will be focusing on you and your slides.
All right.
So I'm very happy to be able to introduce now Jordan Farmer, who is a chemistry major with a minor in computer science. And Jordan is going to be talking about some work with Professors Atamian and Gartner on the identification and quantification of secondary metabolites in, I'm not sure how to pronounce it, so I will leave Jordan to
Pignut.
Pignut. Okay.
Very easy.
All right, pignut. I was not sure, but I'm interested to learn about what pignut is, as well as its identification and quantification. So take it away.
Hi guys, my name is Jordan, and as we just heard, I will be presenting on the plant pignut. So a little bit of background research was a little bit of background information about my research. The plant that I focused on is formally known as Hyptis suaveolens, which can also be more simply known as pignut and also chan. Here's an image of it. It belongs to the mint family, more formally known as the Lamiaceae family, which also contains plants like basil, lavender, thyme, oregano, sage, mint, rosemary, all those nice herbal like plants that we would use for cooking and such. It's a bit of a hairy plant. It has a fuzzy square stem and fuzzy leaves, which are soft to the touch. And it has a minty smell indicative of its inclusion in the mint family. And this grows stronger when it is macerated. It's a plant native to South America and Mexico, but has also been found in more recent years in the tropical regions of Africa, Asia, and Australia. But in its native regions, it is typically cultivated in small village, and it is used medicinally in these villages because of the vast range of diverse secondary metabolites that it contains. A secondary metabolite is a compound produced by plants that don't directly aid in growth or development of the plant, but aid more so in its survival by repelling pests and attracting pollinators. In comparison, the primary metabolites would directly help with reproduction, growth, and development. So some of the secondary metabolites in pignut have been proven to be antimicrobial or act as antioxidants. And the seeds are also used in beverages similar to the chia seed and its beverages. It is native, as I was saying, to South America and Mexico, but it is not useful outside of those regions. In fact, it's actually considered to be a weed in the rest of the world. It's very invasive, beats out native flora, and causes eradication, and is typically found growing on the sides of roads or in undisturbed areas where it can grow to be very thick and bush-like. So with this research, a few broad research goals were developed as a sort of roadmap early on. The main overarching research goal was to develop a protocol for using gas chromatography mass spectrometry to identify the secondary metabolites that were present in our Hyptis samples. The goal was then to compare data that we obtained from the GCMS to already published literature to look for similarities between our samples and research that had already confirmed which compounds were commonly present in this plant. And once we had a broad idea of which compounds we were working with, the next research goal was to make relative quantifications between the various samples that we had about which secondary metabolites were present in more concentration or less than the other plant samples that we had. This would also allow us to determine if various accessions, and I know I'm saying that word a lot, accessions are plants that were obtained from one geographical location at one time. So they're all individual samples of the plant, but they're all from the same species. And we wanted to determine if the certain accessions had compounds that weren't present in other ones to show some type of difference between secondary why did my slide progress? That's weird. Secondary metabolites present in by geographical location. And we had some geographical location data, but not too much, but we wanted to see if we could determine if regions like mountains or found along a riverbed, coastal regions more inland, if any of these geographical factors would contribute to why a certain secondary metabolite would be present or not, and what their concentrations would look like. So to begin, we had some plants. There were a total of roughly 46 different accessions of plants from different regions of Mexico and South America, but we had access to 23, and we grew these in a greenhouse in Irvine for approximately 2 months before moving them over to Keck so that we could easier access them during COVID. And of these 23 accessions that we had, when trials were run on them, we would run 4 different samples from each plant, so 4 biological replicates, and then with these replicates, we ran those 4 times as well for 4 technical replicates, just for repeatability purposes. And the establishment of our working protocol actually required quite a few trials. Prior to protocol determination, a miniature literature review was completed to compile a list of successful essential oil extraction protocols from this same plant. And we kind of compiled from there some things that we wanted to try to see if it would work out. And in the initial testing, we would collect 5 grams of leaves from the plant, store these at negative 80 degrees Celsius if they weren't going to be used immediately in tinfoil. And once we wanted to use them, we ground them up with a mortar and pestle to release the essential oils by slowly adding in DI water until 80 milliliters had been introduced. In this first image, it's just an example of that. You can see there's kind of a lot of leaves still in there. They didn't really combine with the water too well. And this actually resulted in a lot of issues with the beginning of our experimentation. We just had soggy, mushy leaves in our distillation bulb. So that was one of our first trials and errors that we had. We chose to do our essential oil extraction via steam distillation, which we adapted from an OChem lab provided by Dr. Lieberman Martin. And once we put these leaves in the bulb, we were having issues with thorough mixing and heat distribution. And I also had a bit of, I learned that mineral oil, which is the best option for continual thorough heating of a solution, takes a really long time to heat up. So this kind of lab work would take a long time, like took 2 hours just to get the oil to be at the temperature we wanted. So it's a bit of a long process and a lot of learning. So we went ahead with the second test and increased the mass of our leaves to 10 grams because in initial trials with the GCMS, we were not getting any distinct peaks because of there being too little of a concentration from the leaves and the fact that the leaves weren't completely macerated. But we did still have some complications where the leaf tissue was not able to mix with the solution. So we took a different route for preparing our distillation samples. Instead, we stored our leaf samples on dry ice, chilled a mortar and pestle with liquid nitrogen, and crushed our leaf up after giving it some liquid nitrogen so that it became a fine powder. We did add 50 milliliters of DI water so that the concentration would be a little bit higher, and ran into some more technical issues where our solution started to become a slushy because it was so cold. So we allowed that to melt, but oh, my slides keep moving without me. But we were able to successfully perform extraction at this point. The second image is this is with our final protocol. You can also see the liquid nitrogen in the background, but this is where the solution was much more uniform. There were no leaves left in there. And so with distillation, we collected 20 milliliters of distillate and did extraction with hexane to remove the organic layer, and dried the essential oils with sodium sulfate before transferring to a GCMS file. And we performed quite a bit of data analysis. In this research, it became a lot more technical. The raw data from the GCMS came with information including retention times, height, area, and type. And I still don't quite know what type is, PKTY, but I hope to learn a little bit more about it in the future. But we were most interested in the retention time and the peak area from our graphs. So we did a lot of manual work to begin with, going in through Excel files and removing anything that we thought wasn't important. And we have thousands of lines of data. So this was a little bit tedious, but we were able to start to automate this using RStudio to use retention times as an anchor point to merge files together, because each file came through with just one of our samples. And in our initial trials, we had 48 different files to merge with. So we went ahead and merged by retention times, which gave us a really nice, you can see where things came up, where one plant might have had a compound that you don't see in any of the rest of our trials. So we were able to look at a glance and see if there were any major differences right off the bat that we wanted to do further research on. It was really evident that there could also be some machine errors, because with these technical replicates, we would expect that the biological replicates would see some of the same types of peaks at various retention times. And there were some unexpected results, but we were able to move forward with the analysis. We chose to sum up the total number of hits for each accession, or how many times any area was associated with a retention time, to pinpoint times that were common among all accessions, only in 2, only in 1. I don't know why my slides keep doing that, but we'll live with it. And from this point forward, we were able to do a one-way ANOVA statistical analysis, which I have some screenshots of on the bottom of our data merging, but we did continue more with R to look for statistically significant differences in the areas of our GCMS peaks, which would therefore correlate to the concentrations. So for our results, this is just a smidge of what we have. There's a bit more that we didn't get able to look at in as much detail as I would have liked to, but in our early trials, we tested with just 3 of our plant accessions, accessions 1, 4, and 42. And this graph shows retention times that were common among all 3 of those accessions. So we had 8 retention times that showed up in all 3 of these plant samples. And the relative percent area here is proportional to the GCMS of like the relative concentration of the compounds. And from our ANOVA statistical analysis, we obtained statistical significant differences of the means, which was anything with a p less than 0.05. And among these 3 accessions, we found that only our accession sample 4, or the middle orange bar, had any statistically significant differences from the other 2 plant samples. And we marked these statistical differences with a B. And some of these differences seemed quite extreme, such as that corresponding to the retention time of 46.2 minutes, where we are an entire order of magnitude greater of a relative percent area, but others had less significant differences, but they were still statistically significant, such as that at 5.9 minutes. So I went ahead and pulled out, using the mass spec database, what compounds were highly correlated with these retention times and with the mass spec data. And 2 of these were our terpenforol, which corresponded to this first little mini peak of 5.9 minutes. And this is a compound found in tea tree oil, which has been found to inhibit the growth of various cancers, but it does not have any established clinical uses yet. And oh my goodness, I don't know why it does that. And the other compound that we pointed out was at this one where we see the really large difference in peak areas, and that's caryophyllene, which is a compound found in cannabis sativa, black pepper, cloves, and has an anti-inflammatory properties, which we can connect back to how those communities and villages in South America do use these properties, these plants, for medicinal purposes. So we kind of also wanted to look back and look at what the research of what these communities were using to see if we could pinpoint what compounds might be contributing to these medical factors that these communities wanted to use. And we didn't get to complete too many further trials, but we were able to run concurrent samples of 10 different accessions a little bit later in the research. We weren't able to identify what specific compounds were present, but we were able to develop a pie chart showing how each accession, how many compounds were specific to just that plant. So 2 of our 10 did not have specific compounds. All the compounds they had were common, but 8 of them actually had compounds that were present only in that one plant. And although we weren't able to, I do believe that the rest of the research group in Dr. Tommy's lab will be making geographical relations between why certain compounds may have been found in these plants, but not in others. So I do wish I had a little bit more qualitative and quantitative data to show you guys, but this is all I have, and I'd be happy to take any questions. And I do have some works cited, but they keep wanting to move away.
Thank you so much. All right. Let me pause here, give people a chance to please put some questions in the chat or get ready to unmute with a question. Let me stop talking for a second in case anybody has a question now that they want to ask. Oh, Dean Lyon, go for it.
Hey, Jordan, that was pretty awesome. Thanks for sharing. You mentioned that one of the isolates has known anti-cancer activity, but isn't actually in clinical use. Do you have a good sense of why it has not advanced to clinical use?
I don't. I haven't looked at it in a while. I do know they were just testing it among, like, it's been shown against various cancers, so I think they're still looking at it more in a broad in a broad aspect before they would apply it towards any specific cancers and clinical uses. But I do know that a lot of it's in tea tree oil, and I know tea tree oil is a really common remedy that people use for a lot of things. I know it, obviously I don't think tea tree oil would go out and cure any cancers, but I do think it might still just be really early. I looked at this about 6 or 8 months ago, and at that time there wasn't any clinical uses, but I think that it could still be. Also, just there's so much other stuff going on right now, but I do think it would be really interesting to see if it does further and how we can use these compounds against the various cancers.
Great, thank you.
A question from the chat. What interested you to do this research?
So it's more so the fact I was really interested in the instrumentation of using the GCMS. I'm not extremely interested in biology. So the first half of this project was a lot of learning new things and learning about what biological stuff there is. But I want to pursue a degree, a career in forensic science. And I know that GCMS is one of the tools that's really common in forensic labs. So I was just more excited to get more hands-on with the machine and the type of data that it provides and be able to kind of get something under my belt and be able to get a little bit of experience with that. So it kind of just fell into my lap that it happened to, I mean, I got to learn a lot about biology, which was something I didn't expect to do coming into Chapman, but it was nice to have that opportunity.
Nice, fantastic. In the realm of GC, did it take you a while how hard was it to figure out like your run, like exactly the temperature ramp, how to separate everything out?
It did definitely take a while. We did a lot of trials, like obviously our first trial didn't come out with anything. And so we were like, okay, we need to change this, change that. And these runs took like days to do with how many samples we had. Mm-hmm. So we just kind of got lucky that we wanted it to be slow, but not too slow, because then we'd had too much data to go through. So it was a little bit of trial and error with that, but it was definitely, it could have been longer. And I think we got lucky with getting it to be, to start working better for us right off the bat. Nice. But it is a lengthy process. I know these trials were taking like 48 hours to go through like everything we wanted for like
That's a lot. And one other question from your GC data on the next page. Did you, in case I can't remember, did you mention, do you know what that peak at the retention time of 91.2 is, or is that a mystery?
As of right now, it is a mystery. That's something that we want to look more. We weren't able to, from the mass spec data, there were the percentage of confidence that we had for some of these compounds was relatively low. And since we didn't get to run as many trials as we would have liked, I didn't feel comfortable in saying, oh, it's a 40% confidence matter. Let me go ahead and say what this compound is. So if we had had a chance to run some more trials, we would have been able to figure it out. But I think the main thing that the rest of the research group was going to look at moving forward is why this one plant sample had concentration so much higher than everything else, because something that we even found was that this had the smallest leaves. So I was like, is it more just highly concentrated because the leaves are so small? Or we wanted to see if it was a regional thing, many different factors that we weren't sure yet which to pinpoint towards it being the contributing factor.
Yeah, that's really interesting. Very cool. Well, let me stop here and ask people to join me in unmuting or using an emoji to congratulate Jordan on a fantastic presentation.
Yeah, Jordan.
Woo, good, Jordan.
Good job, Jordan.
Way to go!
Yes, thank you, Bryn. And let's see, Veronica, can you get your screen share started?
Yes. Can everyone see my screen?
Yes, we can. And can you still see me if I give you an emoji at 10 minutes?
Yes.
Okay, fantastic. Then it is a pleasure for me to be introducing Tack, Kyung, Veronica Kim today. Veronica is a chemistry major with a minor in secondary education. And Veronica will be telling us about some work with a hybrid cyclic linear cell penetrating peptide. Please take it away.
So hello, everyone. Before I start my presentation, I want to thank Chapman University for giving me this opportunity. Today, I'll be talking about my capstone project, which is about designing and synthesizing of hybrid cyclic linear cell penetrating peptide. So here's my outline and the summary of my research steps. At first, I'm going to start with some backgrounds of my research and the challenges that I had. Then I'm going to talk about my hypothesis that I came up with. And then I will explain some specific aims that with the methods that I used. And lastly, I'll be sharing my results. So here's some background about cell membrane. Cell membrane is composed of mainly of phospholipids, which separates the interior of cells from the outside environment. They're also negatively charged and extremely hydrophobic. The properties of cell membrane create a major obstacle in intracellular cargo delivery. So many biological active agents, such as macromolecules and some hydrophilic agents, cannot appropriately reach the site of the target and be efficient. So in order to overcome this challenge, we need to have a promising drug delivery system. And since the cell membrane is hard to get into, we have to overcome that barrier. Going more, the drug delivery system that I'm going to emphasize is on the cell penetrating peptide. It is a short positively charged peptide with 5 to 30 amino acids, rich in lysine and arginine, which can interact with negatively charged phospholipids of cell membrane and can penetrate into the biomembranes to improve the cellular internalization of therapeutic agents. And we can classify with 3 different CPPs, and the trimeric CPP are composed of 2 or more motifs from dissimilar peptides. And this is the peptide I'll mostly be focusing on. Going more, talking about the cell penetrating peptide. Cell penetrating peptide plays an important role in cancer therapy and is also used as cargo cargo for delivery of peptides and can be used for delivery of imaging agents and radionuclide to cells and tissues. And CBP is also conjugated to the liposome, which the drug was entrapped inside it and can use for delivery of drugs. Okay, so overall, CBP can improve the internalization of the therapeutic agents. It is easily synthesized and functionalized, biodegradable, and easily eliminated from the body. And based on previous studies, it has shown that they have low cytotoxicity compared to the other drug delivery system. Beside the ability of cell-penetrating peptide to transfer the several therapeutic agents, they also have some limitation associated with linear cell-penetrating peptides. And some limitation that it has is it has low cell toxicity, which decreases the desire of therapeutic effect. And they're more prone to endosomal uptake, and they also have poor site stability. So to overcome the limitation of linear cell-penetrating peptide, the cyclic cell-penetrating peptide was introduced, and beside improving stability by cyclization. So many studies have shown that cyclic conformation can scrape endosomal uptake. These are some advantage of having cell-penetrating peptide, cyclic cell-penetrating peptide, and they have higher stability against proteases. They increase cell per membrane compared to their linear counterparts and exhibit efficient endosomal escape. So this peptide that is alternating hydrophobic and charged residue from 1 to 5 in the ring and the tail attached could improve the cellular uptake. So this is the peptide that we start with, and we built it up. And this was the final peptide that we got. And the hypothesis that we came up with is hybrid cyclic linear peptide containing arginated positive and hydrophobic amino acids will act as a cell-penetrating peptide and can be used as a molecular transporter. So we basically said that our peptide, which is this one right here, will have a better molecular transfer than the origin peptide that we worked with. So we have some 3 specific aims that we focused on. The first aim is designing and synthesizing our peptide. The second aim is evaluating cytotoxicity and molecular transporter property of cell impermeable compounds. And aim 3 is determining the cellular uptake mechanism of the peptide. So let's start with aim 1. Aim 1, we start synthesizing a peptide using the resonance of chlorotritol, which already had a arginine attached to it, and because of the time, I'm not going to talk about the whole process. Roughly, the whole process, adding each amino acid, which is arginine, and tryptophan, took about 3 to 4 hours. And it was synthesized with using various coupling by using HBTU, DIPEA, and 20% piperidine and DMF. And all the side chains protecting the group was removed by TAF. And all linear peptides were synthesized through FMO solid phase peptide synthesis method. And cyclic peptides were further prepared by a reaction between N-terminal and C-terminal of the linear peptide in the solution phase. And then, of course, while we're doing this, since our peptide is very long, we need to check the mass if we're doing everything correctly. So we use the MALDI to check our mass. And as you can see, in the MALDI spectrum, the mass that was obtained was very close to our calculated mass. So this is the way we knew that we were synthesizing our peptide correctly. And then after we made our peptide, we need to check for the cytotoxicity is because if you're taking a drug that is toxic, then you're going to have a lot of side effects and it is not good for you. So we had a cytotoxicity that was done in 3-hour incubation. And as you can see, the 10 micromolar concentration had cell membrane more than 90% for all the sequences. So our conclusion was 3 hours incubation and 10 micromolar concentration were selected for cellular uptake studies. And CCRF stem cells and SKOV3 cells were used because those are commonly used cancer cells. So this is another data that we had for SKOV3 cells. And also, as you can see, our 10 micromolar was the cellular uptake study that was chosen. Going on, we also did the cellular uptake studies using FACS technique analysis after 3-hour incubation of the fluorescence labeled phospholipid and the cyclic peptides in CCRF and SKOV cell lines. The y-axis, as you can see, shows the fluorescence intensity and the phospholipid that was used as a negative control, and the regular hybrid peptide that we were used. That were used was effective. So comparing to the peptide that we start with, if we see the FGPYEEI, and when we attach to our peptide, which is right here, it had a very large cellular uptake. So going on, we also did mechanistic studies, and this was determining the penetration mechanism in the presence of endocytosis inhibitor. And endocytosis have 4 different pathways: clustering mediator, cathepsin mediator, micropenicillin, and the chlorine clustering independent endocytosis. And the cellular uptake of the physical mixture of fluorescence labeled phospholipid and hybrid cyclic peptide, which we have right here, were inhibited by chloropromazine endocytosis inhibitor after 3 hours of incubation, and suggesting that the uptake through the cathepsin mediator endocytosis pathway. Lastly, we did an experiment on the confocal microscopy. Confocal microscopy is a microscope that is used to visually see the cells using the fluorescence. And this was done in a 40 times microscope. And the DAPI right here on the left shows the blue nucleus. The Texas red shows membrane and cytoplasma. And FITC is the fluorescence. And like you can see on no treatment, there's no fluorescence at all. And once our peptide is shown right here, there's fluorescence that is shown. So we can conclude that the mixture with our FGPYEEI and our peptide after 3 hours incubation, most of our cells were interacting, as you can see on the merge image right here. So in conclusion, we found that our peptide was significantly more efficient as a molecular transfer than the original peptide and the other hybrid peptides, suggesting that this peptide can be used for more efficient delivery of cell impermeable biomolecules. And thank you so much for everyone listening to my presentation today. And thank you to Saur and Dr. Perrin.
Thank you so much. Fantastic. Let me pause here for just a second, give people a chance to write some questions in the chat if they're currently writing, or does anybody have a question ready to go for out loud to get us started? Oh, Dean Lyon, go for it.
Thanks, Allegra. Veronica, really, really beautiful. When you talked about the synthesis of the cyclic peptide, you mentioned that you release it from the resin and then you do the cyclization in solution. Do you have to do that at very high dilution to avoid combination of multiple linear peptides into larger rings, or is there something else special about that synthesis that allows you to do the cyclization?
So are you asking why we do cyclic synthesis instead of just leaving it linear?
No, I understand the purpose of the cyclic peptide, but when you make a cyclic peptide in solution, the concentration of the peptide matters, because if you have too high a concentration, you can get two linear peptides coming together into a larger ring, as opposed to a single peptide turning into a smaller ring. And sometimes you do that at high dilution. I was wondering if you had particular synthetic strategies that allowed you to get high yield.
That is a very great question, but I never thought about it before that way. I work with one of the grad students, and we basically used what we already had based on other grad students that work with, and then we came up with a different hypothesis of expanding the tail of our cyclic peptide will have better molecular transport. So I'm sorry, but I don't think I can answer that question.
No, no worries. It's not an easy question. Thank you.
Thank you.
Any other questions? If not, I'm curious what kinds of quantities of these peptides you make. It seems like it would be very, very hard and expensive to make them. I'm curious, do you make them in just like really small quantities, or what types of amounts are you working with?
So every time you synthesize your peptide, you're putting more amino acids every time. So usually after we're done making it, if the tube is like this big, we have like this much of our usually solution that contains. But like I said, we have a very large peptide. And just synthesizing one, maybe if you start with arginine and moving from arginine to tryptophan, it takes about like 4 to 5 hours just to synthesize one peptide. So thinking that it takes a lot of time.
Yeah, it's definitely a synthetic challenge for sure. Any other questions for Veronica? If not, please join me in unmuting if you can and giving Veronica a round of applause. Congratulations and great talk.
Woo! Great job, Veronica.
All right, our last talk for this session will be Roxanne at 10:40. Let's take a quick minute to get up and stretch if you want to, get a drink of water, and then Roxanne, I'll have you start your screen share in a minute. Does that sound okay? All right, I will go for a quick walk myself, and I will be back in just a minute. I'm muted. Roxanne, can you set up your screen share? Looks good. Can you see me if I give you an emoji? as a warning?
Yes, I can.
Okay. Then let me just wait another 30 seconds in case people wandered off. I want your audience to be back for the last talk of the session, the first part of the session, I should say. All right, so for our final talk of session 1 this morning, I am pleased to introduce Roxanne Naumann. Roxanne is a chemistry major with a minor in music, and she's going to be talking about some work in my research group looking at lactide polymerization catalysis. All right, go ahead, Roxanne.
Thank you. This is carbodiphosphorane catalysts for lactide polymerization. And it's okay if some or none of those words meant anything to you, because all I really have to say to you today is that plastic sucks. Really. Normally when I make this joke, I can hear you all laugh, but since I'm alone in my room, I'll have to just assume that I'm as funny as I think I am. You will at least be wondering why plastic sucks. And I'm going to tell you, but first I should probably inform you of what a polymer is. A polymer is made up of repeating units of monomers. You put them all together and you make poly, whatever the monomer is called. So here is polystyrene. It's made up of the repeating unit monomer styrene. It's derived from petroleum, which means it comes from a non-renewable resource, which means it sucks. Polylactide, on the other hand, is made up of the repeating unit lactide, which is a known biodegradable polymer. However, it still sort of sucks. Catalysis is an extremely important business in the world. Many things can't be made without catalysts, and polymers are no exception, even the biodegradable ones. The entire goal of the Lieberman Martin lab is sustainable catalysis. Catalysts also sort of suck in that they're made commonly from precious metals. Precious metals are non-abundant, they're toxic, and they're expensive. So what Dr. Lieberman Martin and I have tried to do for the past 2 years is find catalysts that are made out of non-metals. This means that they will be more abundant, less toxic, and less expensive. Unfortunately, things made out of non-metals aren't very good catalysts a lot of the time, so it's a tricky business. Luckily, we've been able to identify 3 really promising catalysts for lactide polymerization, and these include a phosphorus ylide, a cyclic carbodiphosphorane, and an acyclic carbodiphosphorane. And if it holds true that all great scientific discoveries have to be made, at least in part, on accident, then ours is at least in the running, because our initial use of the cyclic carbodiphosphorane came from another lab member who had synthesized it, and our boronic acids and our silanols weren't working as hydrogen bonding catalysts. And we said, hey, this is made out of non-metals. This looks like a nucleophile. Let's try it. And it worked. It worked really well. So to reiterate this reaction that I've been working on the past 2 years, we take our monomer, which happens to be lactide most of the time. We take an alcohol initiator that tells the polymers where to start and finish building. And we mix it with one of our 3 catalysts, and we see how much polylactide we can make. Some key results that I'd like to point out to you today includes 100-to-1 concentrations of monomer to alcohol trials for 30 minutes. We were shocked to see nearly 100% polymerizations for the cyclic and acyclic carbodiphosphorane. Less enthused with the 60% of the phosphorus ylide, but if you gave it 2.5 hours to react instead of 30 minutes, we saw great polymerization yet again. These results were so encouraging that we tried this in a 200-to-1 ratio, that is more lactide and less catalyst, which is good to use less products. We doubled the amount of reaction time, and we saw nearly 100% polymerization for the acyclic carbondiphosphorane and the phosphorus ylide. Unfortunately, the cyclic carbondiphosphorane dropped, but if we gave it 2.5 hours, it got back up there yet again. You might be wondering how we know the yields for these reactions, and we characterize it with proton NMR. On the left, you can see an NMR of just lactide that is unpolymerized. There's a characteristic peak at just above 5 ppm that disappears when it turns into polylactide. So on the right, you can see a graph of polylactide and lactide, and polylactide's characteristic peak is at 5.2 ppm. So if we integrate under both of these curves and we set it equal to 1, we can find the relative ratios of our initial product to our initial reactant to our product. When we try this reaction without our alcohol initiator, things get a little trickier because the polymers no longer know where to begin and finish building themselves. We tried it in a 100-to-1 ratio when we gave it lots of time to react, and we were astonished to find that both the cyclic and acyclic carbondiphosphorane still had nearly 100% polymerizations. The phosphorus ylide fell behind, but that's okay. Another analysis technique that we used was gel permeation chromatography. And a GPC is basically like a giant coin sorting machine, but for polymers instead of currency. Larger polymers come out of this machine faster than smaller polymers. So we saw that when we had an monomer to alcohol ratio of 300-to-1 versus 100-to-1, the 300-to-1 ratio did indeed create larger polymers. When we analyzed this reaction with and without alcohol, we saw that polymers made with alcohol had a very narrow dispersity, shown by the blue, almost looking bell curve on the right. This means that almost all the polymers were around the same length. But when we tried it without alcohol, we saw less of a standardized bell curve shape, meaning that the polymers varied in length more. Overall, I would love to reiterate that all three of these catalysts are actually pretty incredible, and they show huge promise for the future in catalyzing the nucleophilic ring opening polymerization of lactide. The acyclic carbondiphosphorane definitely is the best of the lot, followed by the cyclic, followed by the phosphorus ylide, but they're all fantastic. In the future, we'd like to take a look at two potential mechanism pathways for this reaction, whether it acts as a base to deprotonate the initiator, or whether it attacks the lactide itself as a nucleophile. Our work will continue to be important because it investigates new classes of sustainable catalysts for biodegradable polymers, because plastic sucks and we want to do something about it. Thank you very much to everyone for humoring me in my rants about wanting to save the world.
All right, thank you so much, Roxanne. Here as well, I want to open up the floor for any questions. Does anybody have a question ready at this time? Andrew Lyon, back to you again.
It's a requirement that I ask a question in every single talk. Really awesome stuff, Roxanne. That was beautiful. I love the humor. There's nothing better than edgy chemistry talk. With respect to the polymer characterization, so your GPC curves are beautiful, especially obviously for the alcohol initiated or the alcohol terminated polymers. Do you have any sense of what the actual molecular weights are of the polymers?
No, sir, I do not, because our GPC is currently not working that well.
Oh dear. Okay.
The issue is that I didn't realize this before because I've never worked with lactides, but they have a really low refractive index, especially at low molecular weights like we're making. So the light scattering detectors don't work well. So we need to figure out how to calibrate relative to polystyrene instead. Nobody else will know what I'm talking about, but Andrew, I think.
Oh, wow.
So yeah, so we're going to have to, after Roxanne, the next person on this project will be having to skip the fancy light detectors and do this the simple way, sadly.
That's so frustrating. Okay, cool, thanks.
Yeah, it's a limitation of these polymers that I didn't know of before. A question in the chat, where are you going for grad school?
I'm starting a PhD in inorganic chemistry at UC San Diego this fall.
Very exciting.
I have a quick question for Roxanne.
Yes.
If you could go back to your catalysts, yes, up there. Do you have any idea why because at the end you said you think the acyclic catalyst is slightly better than cyclic catalyst. Do you know why that is?
That's actually a great question that Dr. Ogba asked me on Thursday, and we spent a little bit of time theorizing about it, and I don't have a great answer for you. I really don't think there is a great reason at this point. Dr. Lieberman might know, so it's a great thing she's been here to save me already, but I don't know.
That's okay. Thank you.
Yeah, I think it's still a bit of a mystery to me as well, but lots of interesting things to look at in the future. And I'm so glad you were able to test out so many catalysts so that we can sort of think more about catalyst design going forward. Anybody else? Any more questions for Roxanne? All right. If not, then please join me, if you can, unmute and make some noise clapping and congratulate Roxanne on another fantastic presentation in this session. Woohoo!
Yay! Roxanne!
Pay for plastic sucks.
All right. Well, thank you so much to all of our fantastic presenters this session. We had, let's see, Dela, Jordan, Veronica, and Roxanne. A whole bunch of fantastic chemistry major talks this morning. Let's take a quick break. Cara, you're going to be kicking us off. Can you get your screen share set up?
Yeah, sure. Let me get it.
Perfect. Okay, looks, oh,
Sorry.
There you
Okay.
go. That looks good again. So let me start the second session by introducing Cara Fleener. Cara is a biochemistry and molecular biology major, and she's been working in my research group for about 2.5 years, looking at organic catalysts for ketone hydroboration. So take it away.
Thank you. So you guys kind of got like a quick introduction to my project with Roxanne's intro, so you might see some parallel concepts. So I've been working with Dr. Lieberman Martin with exploring a carbodiphosphorane catalyst in ketone hydroboration reactions. Oh, there we go. So catalysts are really important to study because they're so commonly used in large-scale industrial reactions. 85% of processes that go into creating gross domestic products require a catalyst for at least one step. So they're in really high demand. And luckily, the field of green chemistry has kind of risen to meet that demand by developing more environmentally friendly alternatives to these catalysts. So precious metal catalysts are commonly being used in these industrial processes, but they contain precious metals, and those are expensive. They're not abundant, and they also can cause some toxic byproducts. So instead, my research focuses on catalysts that contain main group elements. Specifically, my catalyst contains phosphorus, and these are environmentally friendly alternatives to precious metal catalysts. So I'm exploring ketone hydroboration reactions, and this is a commonly used reduction reaction in the pharmaceutical industry. It's when a boron-hydrogen bond coordinates to an oxygen-carbon double bond, and it creates this organoborane product, which can be commonly oxidized into an alcohol as well. So generally, these reactions use BH3, which is a toxic and flammable gas. So instead, my reactions use HBPEN or pentyl-borane, but it's not as reactive as BH3. And so we're going to need a catalyst to kind of push this reaction forward. So this is where my carbodiphosphorane catalyst comes in. It's a 6-membered cyclic carbodiphosphorane, and it has a really reactive carbon center because it's electron rich and highly nucleophilic. So this carbon center is bound to 2 phosphorus groups, and each phosphorus group is bound to 3 carbon containing groups. These phosphorus groups are air and water sensitive, so all of our reactions are done in a glove box that has a nitrogen gas environment. So yeah, moving on. I'm just going to talk a little bit about the methods we use to monitor these reactions. We use NMR, and this basically is identifying atoms in our compounds based on their nuclei. And so you can see here for my ketone starting material, this characteristic singlet peak is telling me that these 3 hydrogens are present in my compound. And the first thing I do when I look at my reaction spectrum is look for this singlet. And you can see that it's not present here. So that's my first indication that there's no starting material in my reaction. So furthermore, I can look for this quartet and this doublet to tell me that this hydrogen and these 3 hydrogens are present in my product or in my compound. So this is telling me that my reaction is complete, and I quantify this using percent conversion. So I add an internal standard that's not actually shown in this reaction spectrum, but it shows up as a peak that I can integrate and compare to the integration of one of my product peaks, and that gives me the percent of ketone that's been converted to organoborane product. So this is a general reaction I carry out for all of my ketone hydroborations. Just to note that the majority of my reactions have 1 mole percent catalyst. This is a relatively small amount, and it's done at room temperature. And I reacted a wide variety of ketones, but I wanted to highlight a few steric and electronic effects that I found in my results. So these sterically hindered ketones reacted for a longer time and with a lower percent conversion, even some needing more catalyst compared to these less sterically hindered ketones that reacted for a shorter time and with a higher percent conversion. Additionally, these electronic effects, so these functional groups like bromine, CF3, and CN are all electron withdrawing groups. These reacted faster and with a higher percent conversion than this OCH3 functional group that's electron donating. It reacted for a longer period of time. So these are interesting trends to observe in the data, but they don't really mean anything for us right now, and we can't really deduce the mechanism of action that our catalyst takes based on these results. However, I'm still going to propose the mechanisms. There's 2 possible mechanisms that this catalyst can go through. Sorry, let me just move the people. So this catalyst can react with the borane first through a nucleophilic attack and push a negative charge on the boron, or the catalyst can react with the ketone first, also through a nucleophilic attack, and push the negative charge onto the oxygen. If the catalyst reacts with the ketone first, a lone pair on the carbon center of the catalyst will attack the carbon center of the ketone, pushing electrons up into the oxygen. When the borane comes in, the boron will coordinate to the oxygen, and in our last step, this hydrogen will shift down to the carbon center of our previous ketone, pushing electrons back to our catalyst. This regenerates our catalyst and makes our organoborane product. Our catalyst could also react with the borane first, and this is when a lone pair on the carbon center of the catalyst coordinates to the boron, putting a negative charge on the boron. And when the ketone comes in, the oxygen will coordinate to the boron, and the hydrogen on the boron will coordinate to the carbon center of the ketone. In our last step, electrons will be moved back to the carbon center of the catalyst, and again, we regenerate our catalyst and make our organoborane product. I'd just like to reiterate these are possible mechanisms, and right now we can't tell which one happens in our reactions. So some future directions for this project include exploring different substrates like aldehydes, amines, and drug molecules. Additionally, we could do some kinetic experiments to get some hints on that mechanism of action that our catalyst takes. And so the main takeaways I hope you get from this presentation are that my carbodiphosphorane catalyst catalyzes the ketone hydroborations very efficiently under relatively mild conditions with low catalyst loading and at room temperature. And lastly, the field of green chemistry is just really important to be exploring. I think no matter what field you're in, whatever scientist you are, you should always be open to more environmentally friendly options, whether that be compounds or technologies. So thank you all for listening. And if you have any questions, I'll try to clarify things.
Thank you so much, Cara. Let me open up the chat. And oh my goodness. Okay. Wonderful. I have the chat reopened here. Does anybody have a question to get us started? I'm seeing some congratulations to Cara in the chat.
Can I ask a question? This is coming from a non-chemist, so please bear with me here. When you show two different pathways from one molecule to the product, is there an experimental, and you said we don't know which one it is right now, exactly, we don't know which one it is. Is there experimentally a way to block one to see whether the other works and vice versa? Or is this too simplistic, what I'm saying here?
There's ways to trap intermediates when you're studying organic chemical reactions, but I think because, and obviously Dr. Lieberman could correct me if I'm wrong because this is a little past my scope of practice, but I think these mechanisms are so similar. I don't think either changing the ketone or borane or even the catalyst would be able to trap one mechanism or the other.
So you're saying that you would determine which mechanism it is by looking at the intermediates?
You could, in some organic chemistry reactions, you could trap an intermediate and see, whether this, I don't know, this is just all speculation, whether this intermediate or this intermediate forms or these intermediates. I don't know if that's possible or applicable to this mechanism, but that's been done in organic chemical reactions.
And that's assuming that that intermediate is around for sufficient
long enough to trap it.
Exactly. Yeah. Thank you.
Yeah, always determining mechanisms experimentally is very challenging for sure. A question from the chat from Alex Graydon. Did you look into thermodynamic or kinetic favorable products from your reaction?
No, I think that would be something that would be interesting to explore in the future directions, but I didn't actually explore any kinetics for these reactions.
Thank you. Any other questions? Any more from students? Oh, there's my dog. All right, Cara, in that case, so what do you, I know, but tell everybody sort of what you have planned for after graduation, career goals.
Sure.
Like
so
that.
I'll be working hopefully in a patient-facing field for my gap year, and then I'll be applying to PA school. I'll apply to Chapman's PA school, but also other PA schools in about a year and a half.
Very exciting.
Yeah.
Any other questions for Cara? All right, then let's take this opportunity, unmute if you can, clap, make some noise to congratulate Cara on a fantastic presentation. Woo-hoo!
Good job, Cara.
Great job, Cara.
We need Bryn back. Bryn was making so much noise. All right, we've got a little bit of time, so let's just chat for a couple of minutes. Feel free to stand up, stretch, because we won't start our next talk until 11:40. I want to stay with on the schedule. So let's see. Who else do we have around here?
Since this room is filled with a lot of chemists, for our senior chemists, I have a question for you. So from your experience, your chemistry experience, your 4 years, are there aspects of your program that you would have liked to see that we didn't provide for you? Or new areas to consider as we're always developing and always changing?
I would say that through my research and even more recently when I've been looking at data analysis, I think that it would have been really helpful to have some sort of statistics class or data analysis class to be able to apply to the research that we're doing.
That's a really good comment, and it's been a point of debate, actually. It's a balancing act between adding more units and whether there's anything to remove from the curriculum. So that's a really good suggestion. Thank you.
I also think like maybe if like the FFC like 4th semester thing is gonna keep staying, that maybe it could be like something could be incorporated into that would be like an offshoot. Like that way it's not like too much in the units, but like maybe there's like a few points where it's like this is like a good tool for data analysis, and we kind of just like you focus on it, or you encourage like the students to have some sort of like data analysis in their work. I don't know.
I think the idea of the transition from GCI into the major and that connector, I think is, given that it's a very, it's not been around a long time program, so there's lots to be done in that area. So I agree with you, Jordan, that, having a transition that allows you to see the direction to take what you've learned in GCI is really a good point. More work for us to do.
Was this the year of oh, sorry.
I wish we had aquatic chem.
Yeah, aquatic chem would have been fun.
Were you all the first group to go through GCI?
Yes.
Oh my goodness.
They were the guinea pigs. Nobody wants to say it, but I'll say it.
Yes, we learned a lot from you all. I think this group is also the group that we introduced adding more mathematics into our chemistry curriculum to help with PChem. And that I think we're still analyzing through Dr. LaRue, hoping that was a good addition.
Yes, I agree with that.
Courses.
Oh my God, I agree with that 100%. And we've all talked, all of us in PChem have talked to Dr. LaRue about this, because we're like, we need a little background on coding would be really nice. And I totally agree, because it's the things we've been learning have been so helpful, and I don't want that to leave from PChem. Oh my God, sorry, my dogs. But it would be helpful to have more background, prereq.
So coding and statistics, it sounds like.
Yeah, because I know the bio majors, I think they're required to take Python. So it'd be interesting if that became like a requirement for the chem majors as well. I think it'd be proved to be really helpful.
So if we actually added coursework, would this have scared you away from your major if we added one, maybe even 2 courses here?
No.
Okay, what I have to say in this, I mean, take it how you want, but I feel like having like a coding class, it would be more important than like the linear algebra diffie-q class. And I understand why that one's required, but I personally have not been using that as much as I've been using like the coding and stuff that we learned for PChem at least, so.
Thanks for sharing your thoughts. I mean, this was a great opportunity to get a focus session on some of the, how do you progress and make chemistry an even stronger major than it is?
It's so strong. I cannot express how good the chem faculty is. Like, you guys make the best program ever, and we're so lucky because there's so few of us who are actually chem majors, but I think we have like the best faculty group out of any major ever at any school. So that's my opinion.
I would say we have the best majors of any group, and I'm not even the slightest bit biased.
I think you're slightly biased there, Dr. Short. All right. Well, I think to stay on schedule, thank you for everybody's thoughts. Really useful to hear. I want to let Rose get the screen share started. Ooh, I see some fluorescence stuff already. So it is an honor for me to introduce Rose Albrecht. She is a chemistry major with a minor in computer science. And Rose is going to be talking about some studies of oil products in aqueous solutions today. So go ahead, Rose.
Hi, thank you so much, everyone, for coming. I see a lot of my family here, and I really appreciate you coming to support me. I'm going to be talking about fluorescence lifetimes of oil products in aqueous solutions and their aging and quenching. And I did my research in Dr. Warren de Bruyn's lab of aquatic chemistry. So in order to talk about what my research really is, we have to first introduce you to the problem. The problem is that coastal ecosystems, especially salt marshes, help process natural organic matter, and Southern California intertidal wetlands can experience oil input through a lot of different human-made sources. And the transformation of oil products in salt marshes is poorly understood, and whether oil products are a significant source of organic carbon in these ecosystems is really unclear. And previously in my lab, people were trying to use excitation emission spectra to distinguish between oil-related fluorophores and natural dissolved organic fluorophores, and these EAMs were insufficient. So in order to understand what we're going to talk about next, we need to start with some definitions. First, we're going to look at excitation. When I refer to excitation, that will be the light that causes a molecule to jump from its ground electric state to an excited electronic state. And the emission is the re-emission of light when this molecule relaxes to its ground electronic state again. Our research focuses mostly on the fluorescence lifetime, which is the duration of time between excitation and emission. Also, it's important to note here that the emission will be a lower energy and a higher wavelength than its initial excitation light. So first, when we're looking at our actual data, we're going to see that excitation emission matrices are not quite enough to tell us if oil is present in a sample. As we see on the left, a dissolved oil sample will, when excited over varying wavelengths, which is our x-axis, will fluoresce or emit light at a lower wavelength, peaking around 350 nanometers, and is excited in the range of 240 to 350 nanometers approximately. The problem with this is that a lot of natural samples fluoresce in the exact same range. As we see on the right, when natural samples are analyzed, proteins fluoresce in the same region, and so these scans alone can't tell us if there's oil in water. So what's the solution? We're going to use fluorescence lifetime measurements. Our light source will excite an oil particle in our sample, and our oil particle is dissolved in water. And our oil particle has multiple different components to it that will hold light for different periods of time. Our technology is able to pulse light and detect at what times after the initial light pulse the light is re-emitted. And we see that oils that we were analyzing have three separate excitation emission components with three distinct fluorescence lifetimes. So we're going to look at the nitty-gritty. There's a lot of text on this slide, and if people want to know about the machines that I'm using, they are welcome to read it all, but I'm not going to tell you all about all of it. So we analyzed 13 oil and oil product samples. They ranged from distilled oils like gasoline and kerosene, and they ranged all the way up to crude oil directly out of oil wells. And these were taken from around the world. They were dissolved into a phosphate buffer so that we could get a little bit more oil dissolved into the water. And the aqueous layer was centrifuged out and used as our oil sample because there was oil dissolved in it. Three-dimensional excitation emissions matrices were measured, and time-resolved lifetime measurements were taken. So there's a component with a lifetime around 20 nanoseconds in oil, which is really incredible. As we saw earlier with our three little peak graph, there's oil has a fluorescence component that takes longer to fluoresce than anything we would see in natural samples. In natural samples, the longest fluorescence component that we see is only around 10 nanoseconds. So this 20 nanosecond observation is really incredible. The bulk of my research was to find out if solar radiation affects this lifetime. And the conclusion is no, it does not. When looking at oil fluorescence, component 3, we see that before and after aging, there is a third component peak right around 20 nanoseconds. The lowest measurement that I got both before and after aging was 18.5 nanoseconds, which, while it seems very fast, is exceptionally long for anything we would find in water. So ideally, we could use this technique to take a natural water sample, plug it into our EAMS machine to find what regions excite our oil, and use those wavelengths to find out the fluorescence lifetime of the samples. And if there is a fluorescence lifetime that is around 20 nanoseconds, we can conclude that there is oil in the sample. Another thing that poses a risk to the feasibility of using this technique to detect oil in natural waters is ion quenching. Ion quenching has the possibility to provide relaxation paths that are faster or slower. If it's slower, it's likely to be relaxed so much that there's not a significant light output. And if it's faster, it would not stand out. And we don't know the mechanism at which these ions would quench the lifetime of the oil because oils are so complex and so varied, but we do know they might. So looking at oils dissolved in buffer, when ions frequently found in salt marshes and seawater were added, we saw that only Ki significantly decreased the fluorescence lifetime of our components, especially our third component. It's important to note here that because this was a preliminary study, we don't know the quenching rates of any of these ions, and Ki could possibly not quench the lifetime of our oil at sea level concentrations, as this test was run with Ki at 40 times the concentration that we find in seawater. In the future, I hope to determine the quenching rate of all ions present in salt marshes and oceans to determine if this method of oil detection could be applied to salt marshes and oceans. And as of right now, using like methods, a lifetime component of over 18 nanoseconds could be a reliable predictor of the presence of oil in a sample taken from natural waters. And the feasibility of this method is not limited by the effects of sunlight on oils. Thank you to Dr. De Bruyn for letting me start working in his lab when we couldn't meet together in person and trusting me to not destroy all of his instrumentation. Thank you to my classmate Emma for helping me to really understand what was going on and supporting me through the process of starting in a lab And thank you to all of my chemistry professors for supporting me through the last 4 years. Do you have any questions?
Thank you, Rose. Fantastic. And we will congratulate you again after the questions as well. So let's get started. Does anybody have a question either to put in the chat or say out loud? If not, I have some possibilities. So let me take a look at what I wrote down. Oh, so I was curious, how did you decide on a phosphate buffer specifically? I know so the idea is that it helps dissolve some more of the oil, but did you consider other buffers? Was there like precedent for using that one in particular?
We didn't have a reason for using this buffer in particular, but there were studies done before I started that determined that the phosphate buffer would not quench the lifetime of our oil. So it was a good starting point for the rest of our research.
Got it. Yeah, I didn't even think about that complication with the quenching, but yeah, that seems complicated to figure out. Another question I was wondering about, if you happen to know, what is the current method for detecting oil in natural waters?
From what I understand, it is a just longer series of tests that take a long time. And these machines that we have access to in the De Bruyne lab are expensive and not particularly compact, but they run very, very quickly, and they don't take a lot of training to be able to run. So this method could be used in more varied industries and labs.
That makes sense. How long does each run take approximately?
To measure one graph like this takes about 1.5 minutes.
Oh, wow. Okay, that's really fast.
And when we measured like over a range of different wavelengths, we could measure, say, every 10 nanosecond wavelength emission light, and it could be 30 minutes to measure a whole scan and create data that could show a lifetime peak. So very fast.
Yeah, that is fast. Does anybody else have any questions? I don't want to hog all the time if there's some questions out there.
I had a quick question just on this figure. When you have, and I know this is just a schematic here, but if you have a single step relaxation process, the molecule is going to start decaying immediately after excitation. So with these oils, is there some type of internal conversion of energy that goes to another state, which then fluoresces, which causes a delay in the emission that's shown in this figure?
I don't know, and I think that is definitely a possibility. From what I understand, there are 3 separate regions of an oil that would absorb the light and take different times to fluoresce.
Okay, thanks.
I have a question. This might be a little bit more inside baseball because I use this instrument, but is this data that you're showing, is this all excitation below 300 nanometers, so like at 280 or 270, or did you do any of the stuff in the longer UV, like 340 and stuff like that?
These were all taken with an 298 nanometer excitation lamp. And I think the highest value that we measured was, I think, 350, I believe.
Okay, so you're kind of you're sitting on the peak at the emission peak at 350 nanometers and measuring lifetime. Okay, cool.
Yes.
Any other questions? If not, Rose, what are you planning to do after graduation?
After graduation, I am moving back to Washington, and I will be attending the University of Puget Sound to get a Master's of Arts in Teaching.
Oh, fantastic. That's very exciting. Woohoo! So what age are you hoping to teach?
I'm hoping to teach high school chemistry.
Oh, that's fun. That should be great. And how long is that program?
It's a 1-year program, and I will spend this summer doing some prerequisites for the program.
Nice. Fantastic. All right. Well, please, everybody, feel free to unmute and join me in congratulating Rose on an excellent talk. Fantastic job.
Rose.
Woo!
Good job, Rose.
Great job, Rose.
Nice job, Rose.
Congrats, Rose.
Yay, Rose.
Thank you, all my family, for coming.
Bravo. Bravo, Rose. And all of you, you've been all so interesting.
It's so great to have so much family in here. And it's great to have Bryn back as well. You're good at making noise after a talk, Bryn. All right, we've got a few minutes before Amanda's next talk at noon. I'm going to take the opportunity to stand up, go for a quick walk around, use the bathroom, get a drink of water. I encourage everybody to do the same. And let's reconvene a minute or two before noon so we stay on schedule. All right, see everybody in a few minutes.
You.
Rose, great job on
Bye,
your
Rose.
talk.
Bye-bye, Rose.
It was wonderful. We enjoyed it.
Absolutely.
Thank you guys for coming.
Thank you.
I know.
And thank you, Dr. Schwartz.
Yeah, I just wanted to ask you a quick question. As an undergrad, did you do any preparation to go into teaching, special preparation?
I didn't through Chapman, but I spent 2 summers working for the YMCA outdoor education program in Washington, and I worked with a lot of high school students doing education there.
That's great. I didn't realize with just 1 year master's that you're able to then go into the high school teaching environment. Does that come with a credential at the end
Yes,
of
it
that year?
does.
Wow, that's fantastic. I've always heard programs around 2 years to do that, so that's an interesting program.
So I actually get the credential after 1 school year of teaching, and then the summer of classes afterwards is what adds on the master's from just the credential program.
Oh, it's a great program. Well, I wish you luck with that.
Thank you. I'm really excited about it.
So how much report writing does everybody have to do in the next week?
Quite a bit, I would say.
Just a little bit.
But I think it's easier to like do it after like the fact that like we made these presentations, and so like now you like I feel like I kind of guided my presentation as like I would do for a research paper because it just made the most sense to me. So I feel like it'll make it easier to kind of just power through the rest of what I have to do.
That makes sense. Yeah, you know the order you want to put everything in. You have some figures made already.
Yeah. So I'm hoping that'll kind of just be a nice, easy knockout for the rest of everything.
I also think that by making a presentation, I have fully articulated all of my thoughts on the research that I've done and like how to explain and build the conclusion that I came to in a cohesive way.
Makes sense. Yeah, it's all a little, I think it's a little anticlimactic after this because this is it's so fun. It's like a full Saturday event, and then the report is sort of, it's a good feeling when the report gets turned in, but it's not as public. So this sort of overshadows it a bit, I think. All right, let's see. Amanda, good to see you. You're looking ready with the video on. Can you get your screen share started?
Absolutely.
Okay, it was loading, but it has just loaded for us.
Okay.
and we are just about at noon. So I will, wait, where are you? Oh, there you are, sorry. Everybody's pictures moved around on my screen, but I can see you now, so we're good. It is a pleasure for me to be able to introduce Amanda Waterson today. Amanda is a chemistry major in Schmid with a minor in nutrition. And Amanda is going to be talking about some work looking at brown carbon samples made using a pyruvic acid precursor. Please take over, Amanda.
Perfect. Thank you, Dr. Lieberman. As she just mentioned, I'll be talking about the spectroscopic properties of brown carbon from a pyruvic acid precursor. So before getting into the chemistry and research that I did, I wanted to provide a quick background on its importance and relevance. So brown carbons are a particular type of aerosol, which are solid or liquid particulates found in the atmosphere. These brown carbons have a significant impact on our air quality and subsequently human health. This impact is caused by their ability to absorb and reflect sunlight, affecting our energy balance and climate. Brown carbons have recently gained a lot of attention in the science community due to the lack of knowledge on their exact mechanism and involvement in this climate change. So to gain a better understanding on brown carbon's involvement in climate change and atmospheric impacts, we want to focus on how it contributes to radiative forcing. Radiative forcing is the energy difference between solar radiation absorbed and solar radiation reflected by the atmosphere. The reflection of sunlight by the atmosphere is about 29%, as you can see here, which results in cooling effects. The absorbance of solar radiation in the atmosphere, which is about 23%, contributes to more heating effects. As I mentioned, brown carbon compounds absorb light from the sun in the atmosphere, so we're interested in its absorbance properties and its possible contributions to warming. Brown carbon contributions is one of the largest uncertainties in the global radiation budget, with its net impact largely unknown. These brown carbon compounds account for a large fraction of atmospheric aerosols and have profound effects on air quality and climate forcing. So these brown carbon compounds can be formed through primary emissions from biomass burning, with a large accumulation of brown carbon coming from wildfires. However, we're focusing on the secondary formation of brown carbons from reactions within the atmosphere between volatile organic compounds, or VOC precursors, as seen on this figure, which are aerosols and cloud droplets. These VOCs are oxidized to form carbonyls and ketoacids, which then react with aqueous aerosols like ammonium sulfate and methylamine, which are the focus of my studies. In the aqueous phase, there are many possible fates of these molecules, such as direct photochemistry, oxidation with hydroxide radicals, hydration, and more. This chemistry can also lead to the production of higher molecular weight oligomers, organosulfates, and other organic compounds, in addition to brown carbon formation. So brown carbon absorbs sunlight in the near UV range, which is about 300 to 400 nanometers, possibly contributing to the heating effects in the atmosphere that I previously mentioned. UV exposure also has a photobleaching effect on the brown carbons, as seen in the top figure here, which could change its absorptive properties by altering or removing its abilities to fluoresce. This photobleaching could have an impact on the brown carbon's ability to contribute to heating the atmosphere by decreasing or causing there to be no absorption of solar radiation. So in order to gain a better understanding on the contributions of brown carbon, we aim to characterize its photolytic and spectroscopic properties. This will hopefully allow us to eventually quantify the radiation being absorbed by brown carbon and how it's contributing and to what extent on radiative forcing. So one of the most common volatile organic compounds present in the atmosphere is isoprene. Isoprene goes through many oxidation reactions naturally in the atmosphere, leading to compounds that are precursors to brown carbon. In the first oxidation step, isoprene is oxidized to form carbonyls like methylvinyl ketone. With another oxidation step, dicarbonyls like methylglyoxal are formed. Methylglyoxal is an important precursor that I'll also talk about, and its absorbance properties in brown carbon. Methylglyoxal is even further oxidized to form our keto acid of interest, pyruvic acid. This series of oxidation reactions increases the polarity, which increases its solubility in water. This decrease in vapor pressure and increase in solubility allow these species to react in condensed phase reactions with aerosols in the atmosphere to form these secondary brown carbons. Okay, so I'll explain how we now synthesize the brown carbons from a pyruvic acid precursor with methylamine and ammonium sulfate, which are the two common aqueous aerosols present in the atmosphere. There are a variety of synthesis methods that have been used to synthesize secondary brown carbon in labs, including rotovaping or aging it for weeks or months. However, we wanted to use a method that was simple and could mimic the natural atmospheric reactions of these compounds. So what we did in order to synthesize brown carbon in the lab was we used equal parts of 0.2 molar aqueous solutions of pyruvic acid combined with either methylamine, which is abbreviated as MA, or ammonium sulfate, or AS. These solutions were aged for about a day to fully interact and combine. These solutions were then dried in a fume hood for a couple days at room temperature and pressure to allow for all the water from the solutions to evaporate and the solid brown carbon to form. After drying, there remained this yellowish brown solid that you can see here in the bottom left image, which is the brown carbon. This brown carbon solid was then weighed and redissolved in DI water. This redissolved solution that we have was used to obtain the absorbance and fluorescence spectra of brown carbon and also used in the irradiation experiments to see the impacts of UV radiation on our absorbance. So, like I mentioned, in order to better understand the effects of UV radiation on brown carbon, a solar simulator was used to irradiate the synthesized compounds. On the left here is the exact one that we used. This mimics the radiation from the sun, which is about 300 to 400 nanometers. As we can see on the plot on the right, the radiation below 300 nanometers is not really significant, so only the wavelengths above 300 nanometers will be important in the atmosphere. The samples we synthesized were irradiated for up to 2 hours. Absorbance spectra were taken every 15 to 20 minutes during these irradiations in order to see the changes over time. These spectra were taken frequently in order to get a better understanding of what wavelengths the absorption is the strongest or significant, how strong the absorption is, and how long the absorption lasts, especially with the brown carbon's ability to photobleach. So before getting into the pyruvic acid precursor, we'll quickly look at the properties of brown carbon from a methyl glyoxyl precursor from our previous work. We performed irradiation experiments on brown carbon from methyl glyoxyl precursor with ammonium sulfate. As mentioned previously, pyruvic acid is an oxidation product of methyl glyoxyl, so knowing the properties of brown carbon from methyl glyoxyl could provide insight or allow us to find trends when looking at brown carbon from pyruvic acid. The methyl glyoxyl brown carbon here was irradiated for 90 minutes in the solar simulator, and then its kinetics and absorptive properties were found. Looking at the graph here, between 300 and 350 nanometers, we see the absorbance decrease. But from 350 to 400 nanometers, we can see that it remains almost constant. When the decay at 350 nanometers was focused on, we could see that the absorbance changed as the sample was exposed to UV radiation. As we can see, there's a decrease in absorbance over the entirety of the radiation, and an exponential decay was fit to the data. This brown carbon was found to have a relatively short half-life, around 12 minutes, photobleaching pretty quickly. This could suggest that it accumulates at night and does not persist during the day. Clearly, we see that there's some change in photochemical behavior in these brown carbons. So that covers the basic aspects of these absorption spectra and photobleaching kinetics that we're interested in and what we aim to be able to determine for brown carbons from the pyruvic acid precursor. So now looking at brown carbon from pyruvic acid and ammonium sulfate. In the contrast to the methyl glyoxyl-based brown carbon, the absorbance and photochemical properties of brown carbon from pyruvic acid are much more complicated. On the left graph, we can see that there's a decrease in absorbance from 300 to 375 nanometers, and then it remains constant to 400. On the right plot, we look specifically to extract the absorbance at 350 nanometers to compare it to time. We can see that there's an initial drop in absorbance and then a slight gradual increase. The drop in absorbance is not too large, and each change after is also pretty small. This change in absorbance over time is different from the exponential decay we saw with the methyl glyoxyl. After about 80 minutes of UV exposure, there still remained pretty significant absorbance, suggesting that it's not quickly or easily photobleached, and that its photochemistry is not as similar to that of the methyl glyoxyl precursor as we kind of predicted. Now looking at a different brown carbon compound, it's pyruvic acid and a different common aerosol, methylamine. Looking at this brown carbon, it also shows very different behavior from the other two brown carbon compounds. On the left graph, we're able to see the absorbance decrease at lower wavelengths than the pyruvic acid and ammonium sulfate compound, so from 300 to 350. And then it's pretty constant from 350 to 400. Its strongest absorbance is also at a slightly lower wavelength of 275 nanometers, which is not significantly present in the UV radiation. And then focusing again on 350 nanometers to see that absorbance change over time, as you can see on the right here, there's that same small initial drop in absorbance. However, there's little change in absorbance as the brown carbon continued to be irradiated. This could be interpreted that the UV radiation has little effect on the absorbance and will be able to persist longer for longer periods of time in the atmosphere without losing absorbance. So in conclusion, brown carbon compounds from pyruvic acid precursors show more complex photochemical behavior, but absorption persists for longer periods of time compared to compounds formed from methyl glyoxyl precursors. Future work could be used to further quantify the behavior of brown carbon by studying more complex photochemical models and performing longer irradiation experiments. And then the fluorescence spectra of these compounds could also provide important information on the characteristics of brown carbon compounds and like how their properties differ so greatly between these different precursors. And lastly, I would just like to thank both Dr. DeBruin and Dr. Harrison for their support and encouragement throughout my research and just overall journey at Chapman. And thank you all for your attention. I'd love to answer any questions.
Thank you. Really great job, Amanda. So to get us started today, let me open up the chat. And does anybody have a question to get us started?
So Amanda, great job. I have a question. As you were talking about, like when the brown carbon was synthesized or created, like in the process that you did, was there anything done to try and like characterize or quantify what like compounds were actually formed when that brown carbon formation was done? Or was it more of a holistic view at the whole mixture that you had?
We focused more on just the entirety of the mixture. So including like if there was any secondary products formed, things like that. We didn't try and like quantify if there was, what the different like composition of that synthesis was.
Great, thank you. And just as a quick follow-up, when you talked about like these different brown carbon mixtures that you created, how many different trials did you do with those different combinations that you had?
For each trial, we did for each combination, we did at least 3 trials. And then there was we played around with the pH for some of them as well, trying to see if we tried to bring it to a more neutral pH, if that would change any results. And so we didn't get any conclusive data on that, but we did run many trials on each of these combinations.
Great, thank you.
What was the typical pH for the samples?
They were pretty acidic based off like just the pyruvic acid.
Yeah.
So typically after synthesis, they could be between like probably 2 and 3, I want to say is where we were at. So we try and make them add a little bit of a base to try and bring them up to closer to like 4 or 6. Yeah, what we did.
Interesting. Can you go back to the original structures of the pyruvic acid compared to methyl glyoxyl? If you have those two on. Okay. So, all right. So the kinetics. So it was pretty. Let me know if I'm wrong here. But so you had like the exponential decay with the methyl glyoxyl, and then the pyruvic acid, the much more complicated kinetics where it goes back up. Was that with the same either ammonium sulfate or the other molecule, or were there 2 variables changed there?
I believe there were 2 variables. I think that one was. Oh, let me go forward. There we go. Yeah, this was the ammonium sulfate, and then the one that was increasing. Oh no, that was also the ammonium sulfate.
Oh, okay. Yeah. That's really interesting. Do you know if there's anything in the literature, like are carboxylic acids just more complicated than aldehydes?
I'm not entirely sure. Yeah.
It's interesting. That behavior is so complicated. It looks like the way it goes back up. And this one, you said you did like 3 replications of this one as well, and it just always goes.
Yeah, this behavior was never like any exponential decay or anything linear that we expected.
Mm-hmm. I don't know if it's feasible. Is it possible to get like a time point between 0 and 20?
We could kind of play with any time point. We would just take them out of our Solute simulator and then go take an absorbance vector really quick. So we did some at like 15 minutes, but we typically didn't do anything shorter than 15 minutes.
Makes sense. Yeah, I just wonder like what's happening in between. It would be a bit of a pain experimentally, I think. I don't know how difficult it is to get it like set up and irradiating and then stop it, but I wonder if there's something weird happening in that starting point too. Any other question? Oh, Dr. Schwartz, go ahead.
Yeah, just to follow up on your question, could you have changed the temperature in which you were doing this experiment to actually focus on that beginning time frame?
The temperature at which we were irradiating it or just synthesizing?
Yes. Yeah.
I'm not familiar if we can actually change, because it is in like the solar simulator, if we're able to alter the temperature in that at all. That would be really interesting to see if temperature would have any effect on this.
Do the samples inside the solar radiators stay around room temperature, or do they get like noticeably heated up at all?
They typically warm a little bit. You can kind of feel like the cuvettes get slightly warmer, but they're never like too hot to the touch or anything.
Okay. Interesting.
I was recently reading into the technology behind the solar simulators, and it sounds like the company that makes it advertises that the solar simulator doesn't get above, I think it was 80 degrees Fahrenheit.
Oh, interesting. Any other science questions from students or anybody else? If not, Amanda, I know I asked you this before, but just for everybody else, what are you planning for after graduation?
I'm currently planning on going back home for the summer to the Bay Area, working for a little bit, taking some time off. And then I plan on attending a chiropractic school to become a doctor of chiropractic, probably starting in the January or next spring term.
Awesome. Yeah, I'm so excited. That sounds like it's going to be so cool. All right. Well, in that case, please join me in congratulating Amanda on an excellent presentation one more time. Woohoo!
Woo! Yay, Amanda! Woot, woot, woot!
Yay. All the chem and biochemistry majors are good at making noise and supporting each other during presentations. Bryn. You have such a fancy background, Bryn, by the way.
It's my parents' house. They have a lot of antique books. So
yeah, it looks like a library. It's cool.
Yeah, we call this room the library.
Nice. Well, in that case, let's wait another couple of minutes before getting started with the next talk. Tommy, if you want to test out any technology in the meantime, feel free to. And hi, Tommy. We haven't actually met before. I think I've met everybody else in the room, but nice to meet you. How do you pronounce your last name?
Rocha.
Rocha. Okay. I was not sure if it was cha or ka, so I'm glad I asked.
Yeah, a lot of people get it mixed up because it's spelled like mocha, and so they say it like Rocha.
Mm-hmm. I was tempted to do that. Dr. LaRue, no seniors this year?
Not this year. No seniors.
So strange. Do you have like 20 next year or something?
Yeah, a lot of freshmen, sophomores, and juniors. So.
Okay.
We'll be back next year.
You'll have your own session. Bryn, how are things going? How is PChem Lab?
Well, I will tell you one thing and my students might tell you another, but I've been having a great time.
Nice.
Yeah,
That's
but
fun.
I'm very proud of them. I've like all eight presented today. So everybody's
makes sense.
I'm so proud of them. So
wait, there are 8 people in PChem? Are there people that aren't chemistry majors?
Yeah. So we've got some, we got some biochem students as well. So I love you guys.
Sounds like you're quite popular from the chat, which doesn't surprise me at all. What are you planning for next year?
I'm gonna continue teaching PChem Lab until
very cool.
Yeah, until I manage to, get it together and start applying for things. And I've also been working in a I've been working in a research lab in Orange. They do allergy research. So I've just been kind of like the legwork in the lab there. So
Nice.
been keeping busy.
Cool.
Yeah.
That sounds great.
Yeah.
Well, I'm glad you'll be around in the fall as
Yeah.
well. I'll keep seeing you on the 3rd floor.
Yes. We'll keep passing by. Hello.
All right, we are just about, exactly at 12:20. So Tommy, can you go ahead and get your screen share started? And while you're getting set up, I can start introducing. So I'm very happy to introduce Thomas, or Tommy, it looks like either one, Rocha, who is a biochemistry and molecular biology major with a minor in nutrition within SCHMID. And Thomas is going to be talking about air quality during the COVID-19 shutdown on campus at Chapman University. Please take it away.
Awesome. Thank you. Can you guys see my screen all right? Okay, perfect. Thank you for the introduction, and thank you for all of you being here. Like it was mentioned, I was following, for the past couple of semesters, I've been following changes in air quality here at Chapman due to the shutdown. And so the reason why I was so interested in this is because when you think about COVID-19, you think of all these negative associations, and we're actually presented with a few positive silver linings. And one of the positive silver linings that we have is we're presented with a very unique opportunity as scientists to actually study the influence of human activity, anthropogenic activity. And so this is because, I mean, countries all over the world, to varying degrees at varying times, depending on when the pandemic hit them, have participated in lockdowns. And this has led to an unprecedented decrease in human activity that we've never really seen before, and we probably won't see again, knock on wood. And so we decided to investigate this and have our own little at-home, so to speak, experiment centered around our activity and the implications that it has on our own local environment. I think this is important because environmental issues are generally thought of as a global problem. It's one of those things that's out of sight, out of mind kind of thing. You don't necessarily feel climate change happening every single day. You don't necessarily go to the beach and see water levels rising, but it is happening. And so I think it's important to shed light on how our activity impacts our local environment. And maybe this will bring the idea of environmentalism more to the forefront of people's minds. And the way that we decided to analyze this was through focusing on air quality, specifically in terms of pollutant concentrations and particulate matter. And the hypothesis that we came up with was that due to the decrease in human activity, we should see an improvement in air quality comparing before and after the shutdown. And so our first goal was to set timeframes to compare. The shutdown happened on March 19th. And so we designated February as our data collection timeframe for representing prior to campus closure and April as our after campus closure. And just a brief overview of our design here. Your control would be your data associated with years that are not from 2020, representing normal levels of human activity. Your experimental data would be 2020, given that there is decreased human level activity, independent variable, obviously, again, your level of activity, and your dependent variable being your air quality. And the materials we used to measure these were first the Tropome Sentinel-5. It's a satellite, and it takes absorption measurements and provides that in conjunction with global positioning and weather conditions in order to make inferences and output data about the concentration of common pollutants. And then we also used Purple Air monitors. Purple Air is a very user-friendly online software where you can use freely access data of these sensors. If you look over on the right, you can see what the interface is that you'd be working with on the website. And these are actually the Purple sensors that are all over Orange County. It's actually very popular, which is relatively surprising the first time I used this. And the ones circled in red are the ones that we ended up using in this experiment. And the one in the middle, circled around orange, is the one actually set up at Chapman, which was the initial focus. And down here in the left, you can see there's a model of the particulate matter mechanism. And if anyone's interested in that, you can ask about it later, but I won't get too bogged down in the details of that. So next, this is our process. We didn't necessarily have something concrete that we thought we were going to go through at the start of it, but we did kind of go with the flow and eventually come to a conclusion at the end. So we started off first by looking at common pollutants, and this led us into an interest into Chapman's PM specifically, and this led us into a worry about seasonality. And then we kind of made it come full circle and looked at the types of changes that we were seeing in the data. And I'll get more in detail on that as we go through the results. So again, the first thing we analyzed was common pollutants. And this wasn't necessarily the most definitive analysis that we had, just because only one of the four really showed a change from 2019 compared to 2020, right? A regular year compared to a decreased human level of activity year. So if you take, for example, carbon monoxide, if you look in 2019, in February to April, you see relatively the same trend and relatively the same magnitude of change between 2019 and 2020. And this follows with NO2 and ozone as well. But when you're looking at sulfur dioxide, you finally see a difference. So in 2019, you see a slight decrease from February to April. If you run the sort of significance test, it's actually not statistically significant. The means are essentially the same. But when you look at 2020, you see a very significant decrease in sulfur dioxide levels. And so this actually brought us over to PM, down that PM rabbit hole. And this is because SO2 emissions are largely implicated in automotive activity and construction, which draws a direct parallel over to particulate matter as well, because those are the main emitters of particulate matter. And so we ended up doing the same analysis next with the Chapman PM. And what we saw was all across the board, all the sizes, we saw a decrease in 2020, which seemed at first like a big deal. But then we looked at 2021 and we saw, again, relatively a similar trend is what we thought we were seeing. And so I began to worry. I was like, am I just graphing seasonality? Is that all I'm looking at? Am I just tasked with this Sisyphean task of looking for differences year to year that aren't there because it's just seasonal? And so we went down the seasonality rabbit hole next to try and either support or refute this and see how this played an effect on PM. And so first we expanded the data set, and then to get rid of biases that might be specific to Chapman. And then we plotted it monthly, took monthly averages and plotted that to look at the behavioral characteristics there. And aside from just the eye test where you see differences between 2020 and the regular years, so to speak, you can see, if you look more specifically at the frame that we're worried about, which is February to April, that for the regular years, there is a slight increase from February to April. And significance is not necessarily important here in terms of magnitude, but when you look in comparison to 2020, 2020 has the exact opposite. There's a very significant decrease when you look at 2020. So you're essentially refuting the worry of seasonality that I was having and validating the observations that we were seeing more in 2020 with the initial PM analysis at Chapman. But then I wanted to become a little bit more nuanced in the exact observations that we were looking at when we expanded the data set. And so I went to go categorize the types of change that we were seeing for all the different data sets that were starting to be included in those averages that we just took. And so there ended up being a total of 14 different data sets where we could actually track the changes from February to April. And we categorized those based on, again, regular years or 2020. And this is what I found to be the most compelling piece of evidence, was that when you look at the 2020 data, every single one of those 2020 data points fell in a negative change, meaning that from February to April, there was a significant decrease in particulate matter levels. And then when you look at all the other data points from all the other regular years, you see either no significant difference or you see a positive change, with the exception of 1 point. And this to me supported our initial findings and then also continued to refute seasonality. So when you culminate these observations, we saw that significant decrease initially at Chapman, and then we refuted seasonality, and we further supported those two first observations with what we saw with the types of change being behaviorally uncharacteristic in 2020 compared to all the regular years. And so this goes to show that there was, in fact, an improvement in air quality here at Chapman. And this improvement is associated with the decrease in human activity that took place because of the shutdown. And again, to bring this back into a more general context, right? What I was talking about earlier as environmentalism being a more global issue that people tend to overlook, this is an observable change that we can see in our local environment that we are directly responsible for with our level of activity. And this also has not only implications in environmental health, but also person-to-person health, because the pollutant levels have implications in human health risk as well in terms of cardiovascular health and respiratory health as well. And furthermore, this is not a phenomenon that's specific to Orange County. You're seeing this all over the world. If you look at the Mediterranean Sea over in Turkey, you're seeing species return to those waters that haven't been seen there in years, which I think is very, very impressive. When you look at large cities such as New York City, carbon emissions have decreased by about 50% during the shutdown. And you're also looking at countries as a whole. China is notorious for being one of the largest emitters of pollutants, and we're seeing very large decreases in concentrations of common pollutants such as NO2. So this is an observable phenomenon that's happening at Chapman that is also happening on a global context as well. And further directions that I would be interested in taking this is I would like to see the long-term effects in terms of climate change goals. So implications from things like such as the Paris Agreement. I would love to see how this huge decrease in human activity has actually contributed to, or possibly contributed to, the Paris Agreement, or goals implicated in the Paris Agreement. I'd also like to revisit common pollutants, because the common pollutant data that we were analyzing was being provided by a faculty member that was here. He actually ended up, and we lost contact with him. We couldn't figure out why. And it ends up that he ended up moving to a different university before we got the second round of data. And so I would love to follow up on that and see how that's actually being affected. And then I've become more and more interested in computer science and data analytics. And so I would love to build a forecasting model that would essentially do the analysis that I was doing this whole time, which would be, or what the addition of forecasting. So I'd be able to use past data to understand what's going to happen in the future, as well as understand a standard range, so I could be able to see if there's actually significant deviations at different times in PM. Thank you all for listening, and if anyone has any questions, I would be happy to answer them.
Thank you so much. Really interesting talk. I have one question to get started. So for, you're looking at, let's say, February compared to April. How many samples are taken like in February and how many in April? Is this like a daily thing? And then do you average them? How do those numbers work out?
Yeah, so we took 4-hour averages. You can actually specify like the data that you're collecting from Purple Air when you download it. And so we took 4-hour averages daily, and then we averaged those on a monthly scale. And then we compared those between the different years.
Got it. And was this like a sampling system that was already in place at Chapman and like for other research, or?
So actually this started off mainly as a GCI project. I'm sure Dr. Goldsmith would be very happy about. But yeah, Dr. Harrison was one of my GCI professors, and we initially worked on this in reference to if particulate matter emissions happen from 3D printing. And so we used these sensors indoors, and we did a bunch of 3D printing to see if changes would work, and we did similar analysis. But then when we used it on a more, like a larger scale, we had to do different averaging with the amount of time that we were, or the time frame of the averages that we were taking.
Got it. Oh, okay. So the sensors from a previous project able to be repurposed for Capstone. Yeah. Awesome. Let's see. Any other questions from anybody in the audience? Oh, Rebecca, was that you?
Yes, it's me. Sorry. I'm on a couple computers, so the camera's not going to happen. But I was just wondering what you thought. So obviously, and I feel comfortable speaking for everyone when I say we don't want to do another pandemic again. But what would you say would be policy recommendations that could be built off of some of this research going forward?
So one of the things that immediately comes to mind is when you look at the PM graph of seasonality. Let's see, why don't I just go back to it? When you look at this graph here, you see that there is months where there are like higher trends of particulate matter. And then, like I mentioned before, there are implications in terms of specific health, in terms of respiratory and cardiac health. So in these times where you have higher levels of particulate matter, it would be advisable that you have less activity in terms of like automotive and construction. So maybe you could, I'm not sure in terms of like city planning and infrastructure how you'd be able to do this, but in those months, it'd probably be advisable not to have as much construction. So small changes like that might help regulate these deviations in particulate matter levels.
Nice, interesting. Ramesh, I see you have your hand up. Did you have a question?
Yes. Tommy, there are two questions. One is, did you find any signal of the close by forest fire? And also you showed the changes in the SO2. What are the sources of SO2? I joined this room late, so I could not see that. Okay, so could you answer me?
Yeah, so in terms of the second question, the sources of SO2 are mainly automotive activity and construction. Those are two of the main contributors that we are focused on. And that's kind of what led us down the rabbit hole of looking at PM, which has similar main emissions there. I'm sorry, I didn't hear your first question. Could you repeat your first question, please?
My first question is, did you find any signal from the close by forest fire?
Oh, yeah.
Because yes.
That's exactly what's being shown in this graph here. Between September and December, that huge peak in 2020, that's all from those fires.
Did you look at the wind directions?
No, we didn't analyze that. No.
You should have seen that. Okay. And also, we are very close to Disneyland. Okay. And whenever we are measuring the PM 2.5 on the campus, whenever the fireworks happens, you get a peak. So did you see that in your data?
Actually, that's not something we considered, but that would be something that'd be very interesting to explore, I think, actually.
Yeah, they also stopped the fireworks for the year.
Yeah, because always what I've seen at 10 o'clock, 9:30, you see, you get a peak due to the fireworks in Disneyland.
Yeah, that's something that you can actually analyze on the Purple website by itself, because it does graph those that data on its own. So if we had a sensor over at Disneyland, that'd actually be really, really interesting to see what happens when we track the schedule of fireworks. That'd be actually a very interesting project.
That would be cool. Yeah, somebody should definitely do that in a future project. I could definitely see a GCI project like that. And Brian, I saw you had your hand up. Do you want to give us one last question for this speaker?
Yeah, thank you. First of all, great job, Tommy. Really proud of you. I might be mistaken, but was there an increase in carbon monoxide level between April and February?
Yeah, so there were increasing levels between February and April for 2 of the 4 common pollutants, but those were the trend was exactly this, or relatively the same between the 2 years, between the regular activity levels and the decreased activity levels, and it was relatively the same magnitude as well.
Got it. Okay, makes sense. Thank you.
All right. Thank you so much for all the excellent questions. Thank you, Tommy, for a wonderful talk. And everybody, all of the attendees, please join me in congratulating Tommy on a great job. Woohoo!
Yay! We're done.
Woot, woot, woot,
Good job.
woot.
And in another minute or so, we will get prepared for the final talk of the session from Daisy. Do you want to test out your technology while we wait?
Sure, yeah, I'll start screen sharing.
All right, I see a slide. Now I see a title. Looks good. I want to wait another 30 seconds or so. Let any last people get into our breakout room before 12:40 when we begin. All right, we are just about at 12:40, so I would like to, it's my pleasure to introduce Daisy Haas, who is a biochemistry and molecular biology major with a minor in rhetoric and writing. Today, Daisy is going to be telling us about some organic chemistry, chemical education research. So as an organic chemistry professor, I'm very excited to hear about your findings. Please go ahead and get started for us, Daisy.
Thank you. Thanks for the introduction. So I'm Daisy Haas, and my research today is on utilizing Millie to assess the experiences in Reactivity's organic chemistry labs. So reactivity stands for Reformed Experimental Activities. And so this is a reformed learning pedagogy that was implemented at different universities with the aim of transforming the undergraduate organic chemistry laboratory to improve student engagement, problem-solving skills, and confidence in the laboratory. So the traditional method of teaching an organic chemistry laboratory usually features a pre-lab lecture that centers a professor or TA, and then the student begins the experiment on their own. Reactivities aims to transform this learning experience into a student-centered experience by inviting students to enter the lab and immediately begin with partner work and practice with the materials and methods that they will be using in the procedure. This is meant to develop communication and a community within the laboratory, and then to get students more confident in their abilities to work with the methods and materials before actually engaging in an experiment. You'll also notice these contextually placed stop signs that prompt students to reflect on conceptual material, discuss with peers and TAs, and also begin the problem-solving skills before the lab actually begins. This is also meant to relieve some of the anxiety that students may have when it comes to messing up during an experiment and making mistakes. So reactivities is basically trying to deliver the same material, but a different method. So they're trying to aim to figure out some of the lacking points in traditional teaching methods for the organic chemistry laboratory and really make a difference for students. So one of the items that is maybe lacking in traditional activities is the lack of molecular connectivity. This relates to the connection between the macroscopic view of the laboratory and the molecular view of the lecture. So students may not understand what the molecules are doing and how they're behaving when they're doing techniques in the laboratory. This is maybe exacerbated by the fact that when you enter the lab, sometimes you have a different substrate every week. Reactivities aims to address that by implementing a communal pool of substrates that have a structural motif that is common between all of them, so it's easier for students to track changes when they go through their techniques. So this is actually great because it's cost efficient and it streamlines the process of preparing chemicals for the laboratory. It is also great because it adds some type of recall for the students, as for example, for 1 week they are learning thin layer chromatography and they will be using compounds 1, 2, 3, and 4. And then the very next week they will go through column chromatography and separate compounds 3 and 4. So that sense of recalling the structures that they had used the week before can really help them to revisit techniques and concepts that they had already learned in the lab. Another idea is the disposal of newly synthesized compounds. We're really focused on building confidence in students. And when you ask them to prepare a material for 4 hours and then congratulate them and then tell them to put it in the trash, that's a bit of a bummer. And so we want to build that confidence in students and show them that their work is really important. So moving on to the assessment techniques, when we implement this learning style, how can we tell that it's actually making a difference in students' lives? My research focused on the question of, will students report gains in the affective and cognitive areas when reactivities is implemented? So I assess this utilizing the meaningful learning in the laboratory instrument, which I refer to as MILI. So MILI is an online survey that is looking at those affective and cognitive domains of student learning through an online survey that is delivered before and after the semester of organic chemistry. It's a 31 item survey that includes one indicator item and both positively worded and negatively worded items. So reactivities was implemented at 7 different universities, ranging from aspiring R1 universities to community colleges to smaller liberal arts universities. And so every time we implement a teaching style somewhere, every teacher is different, every student is different. So we are going to get different outcomes when it comes to the cognitive and affective growth of our students. One way to visualize this growth is by looking at the individual questions in the MILI survey through averages. So this top graph looks at the averages after Organic Chemistry 1, and this bottom looks at the differences or the averages after Organic Chemistry 2. These basically look the same. So the best way to visualize the change in growth for targeted assessment is looking at a net average of those individual questions. So one thing I noticed was a lot of these have very small net growth, and that's actually really promising results because those are high scoring questions and they're consistently high scoring. So that means that students report highly every time and it stays consistent throughout their time in organic chemistry. So these questions that score really highly and are consistent include making sure that data makes sense, making mistakes and trying again, learning problem solving skills, and developing confidence. So these are really high scoring and they're consistently high scoring, which shows that problem solving and confidence are both things that are developed within this laboratory style. Now, one thing to point out are these 2 negative net averages in red, and those actually refer to the idea of molecular connectivity that I discussed before. So students report low and they are reporting a net loss between Organic Chemistry 1 and 2 of connecting what the molecules are doing to that macroscopic view of the laboratory. And that's something that we'll want to consider in our future directions to understand why that is something that students are lacking. Another really sad result was unfortunately these 2 net differences. These are net losses that refer to being excited about chemistry and learning chemistry that students think are going to be important. This is sad to me because I love chemistry and so I want students to love chemistry, but it does make sense because this is after Organic Chemistry 2, so students may be burnt out from their time in organic chemistry, so we can understand why that's something that students are reporting, but that is something that we want to improve because we want students to enjoy their time in what they're learning. So I talked about confidence a bit earlier, but confidence is something I'm really passionate about because I think that it's incredibly important to develop confidence for groups that might be underrepresented in the sciences in order to continue their retention in the sciences. So specifically for groups such as women and BIPOC students, confidence development is very important. So in this graph, I'm looking at confidence averages for each reactivity student. So then on the scatter plot, each point represents one student who has correctly filled out the survey. And so this shows, because they're very aggregated up in this top right corner, that shows that students are reporting very high confidence through Organic Chemistry 1. We ideally would love to see all of our points above this line because that shows a net positive, which means that they have grown confidence throughout their time in organic chemistry. But because they're all concentrated up in this high range, we like that. There are very few outliers who reported low confidence. And so that's really great. We love to see, students be confident. Now this graph, which is the negatively coded confidence questions. So I feel unconfident in the lab. I feel scared of making mistakes. Those types of questions, we see that it is very scattered. And so that may point to the way that students reply to survey questions that are negatively coded. And so that's something that we will want to explore in our future directions of doing semi-structured interviews with the students. So semi-structured interviews are interviews in which we ask our students some questions to answer some of the questions that we have about our results from Millie. So that molecular connectivity that we saw, that's something that we'll want to dive into, as well as that lack of confidence. And so that is our next direction. We're currently writing the protocol for those semi-structured interviews to gain more of a holistic view of our students rather than just a point on a graph. Additionally, to conclude, my main takeaway is that Millie shows evidence that reactivities can foster the learning cycle through practice and grow that confidence in those problem-solving skills in our students. And I think that's something that we can implement in all of our classrooms, whether it's reactivities or just more of a student-centered approach. So I would love to thank the National Science Foundation for funding the REU program that I attended and also the research project, as well as my principal investigator, Dr. Gaudreau, and the entire lab at RIT, and specifically at Chapman, Professor Dudley, and Schmid College of Science and Technology. And if anyone has any questions, I would love to answer them.
Thank you so much, Daisy. Really good talk. Let me open up the chat box. See, does anybody have a question to get us started for Daisy?
I have a quick question just because I popped in a little late. What student population were you looking at? Was it at Chapman or I missed that? Sorry, I just was curious.
No, that's okay. I'll go back to my, sorry. So we implemented it at 7 different universities. We only had data on 6 so far. So it's ranging from aspiring R1 institutions to community colleges to smaller liberal arts colleges.
Gotcha. Thank you.
Yeah.
I was curious about did you notice any difference in responses between students at different universities, or was it pretty uniform?
Yeah, we do. In this descriptive statistics table, we do see a difference in the amount of cognitive and affective growth in our students. Specifically at Rochester Institute of Technology, we had a larger sample size, so we were able to see more trends in data rather than some of our smaller universities where we had sample sizes of 10. That's also not just because it's a smaller university, but because we had to get rid of some of our students who filled out the survey because they filled it out incorrectly. And so because of that, we are analyzing trends, but it is difficult when we have drastically different sample sizes to really analyze those statistic differences.
I have a question, Daisy. Do any of your questions address what's known in the organic world as the preconceived notions before you come into, or attitudes before you come into organic, and how does that impact the approach that you're using? Do you see differences, or did you actually assess students that came in excited, students that came in not happy about taking organic, and how that changed?
Mm-hmm. Yeah, that's a really great question. A lot of students can be very scared of entering organic chemistry. And so if you look at this affective growth, so that's referring to our students' emotions, moods, and kind of just ideas about themselves effectively, you can see that the averages for our so that's before the semester starts, are much lower than our cognitive averages for our pre's. So as we can see, most of our averages for pre-cognitive, that's discussing their actual knowledge, so their knowledge growth, they have pretty great, they think that they're going to learn a lot, and they think they're going to do problem solving skills and learn a lot from organic chemistry. But when it comes to these affective averages, they're much lower in the 60s. And that just kind of hints to us that there may be some fear when entering the organic chemistry lab. And so sometimes you'll see growth in this affective growth. And then sometimes you'll see, these are just overall averages, so these aren't individual students, but you'll see some growth, some decrease a bit. And so that's just, yeah, I think that's a huge part of it. We do find that those pre-surveys, depending on when they are given, if they're given right before the semester or a little bit into the semester, that can also make a difference on how much fear there is. So that needs to be streamlined as well in the methods.
Thank you.
Yeah.
There's a question in the chat from Rebecca that Adena seconded about, were you able to stratify any of your data according to gender identification and or Black, Indigenous, person of color status?
Yeah, that's a great question. So that is a question that we added into our survey a few years into implementing it. And so like I focused on competence, another group member was looking at gender. And so race is something else that we want to add into our surveys. In terms of gender, most of these universities were actually female dominant. So they had a bigger percentage of female students. And so that changed the differences a bit just because of that population size. But that is something that we want to explore more, especially in larger institutions or institutions that are more male dominated. And then in terms of BIPOC students, I think that's a really great study that needs to be done. And that's not currently being done, especially because some of these institutions are a bit more predominantly white institutions.
Let's see, Professor Gardner, did you have a question?
Yeah, so great presentation. Thank you, Daisy. I just had a question, like, what did you know if there's any difference between the preparation that these students get coming in organic chemistry? Like they have these reactivities kind of format for their organic labs. Was there any similarities or differences did you look at between what they would have gotten in general chemistry that may have influenced their preparation or their own perceived expectations for organic chemistry? Was that something that you looked at?
That's not something that we looked at, but I do see how that would make a difference. If they had a reformed general chemistry laboratory, I can see how they would come into organic chemistry with also expecting such things. But that's not something we looked at in terms of what they were coming into organic chemistry with, mostly just what they were expecting from themselves. So in the cognitive and affective areas, it was mostly focused on what they were expecting from the semester and then what they got from the semester, rather than looking at like previous education. But that is a great question, and I think that could be something that we focus on in the future.
And one more question from Shun in the chat was, do you have any data on the differences between various majors?
That's also, and so we have some students working on specifically confidence, major, and then what year that the students are in. So if year makes a difference, if you are a sophomore taking organic chemistry, or if you're a senior taking organic chemistry, that may make a difference. And then gender was another one. So we do know that majors tend to, specifically this question, the one where, oh, I'm going backwards. Let me see if I can find it. But the question that referred to enjoying chemistry more, that actually tended to be higher rated among chemistry majors, which makes sense. And additionally, that post-organic chemistry, if I can get there, that has higher scores as well, because mostly the people who take organic chemistry 2 are majors who are focused in chemistry. So they tend to have a bit higher of scores rather than that organic chemistry 1, which may include non-majors and just non-science majors in general.
That makes a lot of sense. Any last questions for Daisy? All right. If not, everybody please unmute if you're comfortable to give Daisy a round of applause for a great talk. Woohoo!
Great job, Daisy.
Good job, Daisy.
Good job, Daisy. Nice job.
Great job, Daisy.
Good job.
Wonderful. And that is actually concluding our second session of the afternoon. So congratulations to all of our wonderful presenters today. I am so glad to have been in this room all day hearing a bunch of great talks from a bunch of chemistry majors and a few biochemistry majors as well. I hope you are all more relaxed now after your talks and ready to socialize a bit for half an hour and close out the conference in the main room.